All right. So what I want to talk to you about today is, you've heard already in a number of talks about, these two-dimensional active pneumatics, and I want to tell you how the dynamics of these materials generalizes to three-dimensional when you go from 2 dimensions to 3 dimensions, right? And it's in a certain way it's non-trivial, right? And this is as much a talk about experimental techniques of how you characterize large-scale dynamics of some millimeter-sized dynamical system with a single particle resolution. Okay, and so this was really done by two experimentalists, a postdoc and a grad student, Dan Beller, who really is an expert on liquid crystals and structure of liquid crystals, and they've done most of the work, right? And so this is the famous instability that was put forward by Sriram 17 years ago. And so this is an aligned microtubules. They are consuming energy and extending, and these aligned microtubules become unstable and develop this instability that grows in this direction. And over time, they exhibit this, they go into this steady state chaotic dynamics. And this is really the phenomena that drives a lot of the rich phenomenology of active materials, right? So in 2 dimensions, for example, and so now what we have is we have these microtubules live on an oil there are quasi 2-dimensional oil water layer. And so the oil is below it, the water is above it. I have the microtubules here, and I have this initial bend instability. And so what the active pneumatic wants to do, it wants to bend more. And as it bends more, right, the stresses build up. And at some point, the field, the pneumatic field that describes the average alignment of the microtubule is no longer continuous, and you develop a fracture line. And this fracture line is terminated by what I will call 2 topological defects. And these topological defects have a charge of -1/2 and a +1/2. And this topological defect goes forward on its merry way. It can also happen that these 2 topological defects encounter one another, they approach each other, the material between them moves away, and you end up with something that's a smooth field as is shown over here, right? And so in 2 dimensions, you get dynamics that looks like this, right? You have these microtubules, you have a pneumatic director field defined everywhere. You have the velocity field that you can measure. And so, for example, here you could see an annihilation event. Here you can see a creation event of 2 topological defects, right? And so they're moving around, they live for a very long time. And it turns out that this is a very complicated dynamics, but the dynamics of these complicated systems to a certain level can be described by the dynamics of these simpler topological defects that are definitely simpler than the entire fields of orientation and velocity, right? Again, this is a 2-dimensional system so far, right? And so these topological defects that have a charge of +1/2 and -1/2, they are distinct, right? And the only way I can create a pair is, by taking via this unbinding transition or by combining them, right? And you'll see that in 3D that actually changes quite a bit. And so this dynamics is definitely seen in, this is a system with the orientational order. There's, this is just a natural way to describe many different systems. So one of the first ones that Sriram mentioned was this shaken granular rods already. You can take a lot of cells by themselves, have anisotropic order, as is shown over here, right? And again, you can use this pneumatic order. And again, pneumatic order is a vector that has no arrows. And you can describe the dynamics of this system. You can describe dynamics of bacteria, of cytoskeletal components. This seems to be a fairly universal description, right? But a lot of these systems are inherently quasi-2-dimensional, right? It's very hard to take granular rods and build a 3-dimensional material, or even cells. If you were to build a 3-dimensional tissue, imaging this tissue would be very difficult because of the scattering. And so, a lot of these things are married to 2-dimensional systems, but what we want to do is what we wanted to study is what happens if you take this material and you make it three-dimensional. The dynamics of a three-dimensional material will obviously be very different from the dynamics of a two-dimensional material. And so that's, I've said, so that's what I want to address in my talk, right? So the first thing I want to say is how we go about making this material and imaging its spatial structure and temporal dynamics. And then I want to say something about the peculiar role of topological defects in 3D active nematics as opposed to 2D active nematics, okay? And so what are our basic materials, right? So again, we do kind of Frankenstein experiment where we take biological building blocks and use them in a completely non-biological matter. And so for example, here I have microtubules that you purify from tissue. These are long filaments that can be microns in length. In our case, it's about 1 micron in length, about 20 nanometers in size. And so these are basic building blocks that make our orientationally ordered fluid. The other basic component that we use are kinesin molecular motors. Again, these are motors that are responsible for a lot of the motion that occurs within the cell. These motors hydrolyze ATP. They take 8 nanometer steps, and they take about 100 steps per second along the microtubule. So they bind to microtubule, they take an 8 nanometer step, and they take 100 of these steps before they unbind again, right? And so the third component that we add is something that's going to bundle microtubules and make these elongated bundle structures that are shown over here. If you put all of these things together, what happens is you have microtubules. The motor, when it lands on a microtubule, it knows which way to go. It moves towards the plus end. And so if a cluster lands on a microtubule, it's going to move in a plus and towards this direction. If this microtubule has the opposite polarity, it's going to move in this direction, and you'll get active sliding that's going to generate locally active stresses. Okay? And this is what you see, right? If you take a lot of these microtubules, and if you increase the concentration, this is the dynamics that you see. Now you have a network of these microtubules. This is 1.5 millimeter length scale. Each microtubule is about 1 micron. And you see these large scale dynamics, these pieces of experimental crud, and they're being advected by this, fluid that exhibits this kind of chaotic, turbulent-like behavior. And so we've been working on improving this. So if you zoom in, this is what's happening on a molecular length scale, right? So this is a very, now this is 100 microns in size, right? And so you can see that this is a very dilute system, right? These are microtubule bundles, and you can see that these microtubule bundles are pushed apart, but because they're connected to the network, as they're pushed apart, they buckle, as is shown over here, right? And they fracture, and then they connect with other microtubule bundles that are shown over here. And so you'll get this cascade of these filaments being extended, buckling, fracturing, and reforming the network. And that's what drives the large-scale mechanics of this material. And, and we have some, so what's nice about these materials, right, is first they're made from the bottom up. So I have control. I can make these motors go slower or faster by changing ATP. I can change their concentration. So I have a lot of control over the microscopics. They're very long-lived, right? So you can go from hours, and actually recently we've been able to implement a new strategy in which the dynamics goes for 120 hours, right? And so, you can really do kind of quantitative experiments. Usually the lifetime of these things is a problem because they ran out of fuel. These things are still temperature fluctuations that data was taken at Brandeis at the lab that had no temperature control. Now temperature at Santa Barbara is very flat. All right, but if you look at this thing, what you will notice is that there's this three-dimensional bulk. I can make tens of milliliters of these things. This is by no means an orientationally ordered fluid, right? I have these fibers, but these fibers essentially are are just randomly oriented. So it's isotropic in every sense of the word, right? But what we wanted to do is we wanted to build something with the orientational order parameter, right? And so what we do, and this took actually a lot of work, is we've taken a passive liquid crystal, right? So we take virus particles that are rod-like, we concentrate them, and at some point they undergo a transition from an isotropic to a nematic phase. In a nematic phase, all of the rods are pointing in the same direction. So you have a birefringence, and you see this birefringent phase at the bottom and an isotropic phase at the top. And then when you combine that, we've managed to combine this, which provides the orientational orders of the freedom, with this, which provides the active stresses that drive the liquid crystal out of equilibrium. This, by the way, is nice because in a two-dimensional system, active stresses are always coupled to passive stresses, so you don't have the independent control of them, right? If I change ATP, I'll change the elasticity and I'll change the active drive. And so we can explore the phase diagram in much greater detail. And I don't want to, bore you about the, biochemistry that we had to do to accomplish this. But this is what you see now, right? This is an active three-dimensional nematic. And if you look under polarization microscope, what you see here is that it really lights up. So initially what you were doing is you were flow aligned in this direction, right? And then it undergoes again this instability, this reram instability in this direction, and then it goes into an active turbulent state. This is 500 microns in size. Each one of my rods is about 1 micron in size, right? And so we were very excited about this for about 24 hours because, now we have our 3D active nematic, and then the question is, well, what do you do with this, right? I mean, this is a mess, right? I mean, so that's not obvious, right? And so what one would really like to do, right, is one would really like to get a full spatial and temporal structure of this image, right? Add a micron length scale, because that's at a length scale of hundreds of microns. But to reconstruct the nematic order parameter, I need to know what each individual rod is doing, right? So I need to image micron rods, and I need to image the large scale structure at almost millimeter length scales. You can try to do that with a confocal, and which is the kind of standard go-to technique, but that's not gonna that simply doesn't work, right? The other thing that we want is we want to image how the structure evolves in time, right? And so that's where our system is very nice, because we can lower the ATP concentration and really slow down the dynamics so that we can study the structure of our material. So what we've actually adopted is, is techniques from the community of developmental biology. So people have actually been able to take a movie of a Drosophila embryo develop into a full fruit fly with remarkable resolution. And so it's quite remarkable. And that's called a light sheet microscopy. So what you do in a light sheet microscopy is you take a sheet of light and you illuminate your sample. You illuminate a very thin 1 micron thick sheet of light, and you image from this direction. And so within a millisecond, you can obtain an entire two-dimensional image with 1 micron thickness of your sample. If you do that with a point scanning confocal, even with a spinning disk confocal, that would take much, much longer, right? And then what you do is you scan your sheet to obtain three-dimensional data, right? And from this three-dimensional data, this is an example of it, right? So this is my sample. I have my three-dimensional active nematic. I have a light sheet over here, and you can see there's dots here. That's the rods pointing into the plane of the screen, microtubules. There's rods lying in the plane of the screen, okay? And this is how the raw data looks like. Okay, what I should say is we do multi-arm imaging. So we have 2 light sheets, right? And you have 2 imaging arms, and that really increases your resolution, where you can do 1 by 1 by 1 micron imaging with the temporal resolution of 10 seconds, where your spatial resolution is sub micron, right? So The other thing that's really essential is if you just, if you image with a lens, your point spread function is going to be very anisotropic. And if you want to extract anisotropic directors, that's a nightmare. What we do here is, because our sample is has a cylindrical symmetry, you can simply rotate the sample and take the sample from multiple images, and that makes your point spread function isotropic, and that makes life much easier. And so if you do this whole thing, what's happening here, this is our raw data, right? This is our tube. Again, this is about 800 microns in size. This is a tube. I'm looking from it, and this is not a temporal resolution. What's happening here is we are scanning along the z direction, and here you are scanning along the length of the tube, right? So you'll see when you come to the edge of the tube, all of the rods are aligned along the tube surface, and then if you go into the bulk, the director changes in all kinds of directions, right? And so you can take all of the scans with about a temporal resolution of about 10 seconds. And we have some ideas about how to improve this by at least almost 2 orders of magnitude, and then we can really get very fast dynamics. All right, if you do that, the first thing that we did is we extracted the nematic order parameter using the structure tensor analysis. And so you can see, for example, these are the red lines. These red lines, again, indicate rods lying in the plane of the screen. And then once you have dots, that really shows that the rods point into the plane of the screen. Once you have the director, this is a 3-dimensional full director field on millimeter length scales, yes. I mean, because we don't discriminate against X, Y, and Z. We have full 3-dimensional data, right? Yes.
Do your filaments have any preferred alignment at the walls?
Yes, you can see that in this previous movie, right? They will want to lie parallel to the wall. You can see here, right, if I go back, these are the dots, that means and then if I go back to the wall, what you'll see is, this is the beginning of the wall. They all line along the wall as, just due to entropic reasons.
So the viruses are what scale?
1 micron as well. Okay, so, so this is 1,000 by 1,000 by 1,000 rods, right? I think in a few years, maybe you can simulate it, but I have a few years. You're what, 1,000 by 1,000 you are right now? Okay, 1 billion, right? Okay, there's still, once you can simulate, there's no use for me as experimentalist, right? Okay. All right, so the next thing that we do is we plot the distortion energy, right? So you have a director field. If all of the rods are aligned along the same direction, the distortion energy is going to be very low. Where the distortion is very large, that's indicated with these yellow lines, right? So that's the large distortion energy, and you can see where you have these rapid variations of the director field, which would indicate maybe presence of some kind of defect. Those are these yellow lines, right? And again, what we are doing here is we are scanning in Z direction, right? And so if you have all of this data, right, you can just do a 3-dimensional reconstruction. And this is the instantaneous structure that's telling you where the high energy distortions are, right? And you can see that the sample has, these green lines that span the entire structure, entire sample areas. And then there's this isolated loops that are shown in red here, here, and here, right? And you can imagine that this is indicative of presence of topological defects, right?
Distortion energy is something like n to
Yeah, it's just gradient of the director field with one constant approximation, right? But moreover, what you have is, you can then reduce the ATP concentration and see how these structures evolve in time, right? And so, for example, that's what happens here, right? Again, our temporal resolution is right at the point where you can start to follow the dynamics of these structures that we observe in 3-dimensional data, right? So, for example, what you can see here is we'll focus on one of these high energy distortion regions. You can zoom in and you can see that this breaks up into multiple loops over time, and these loops evaporate, right? Now we played back in time, and we just reposition this to show that, there's actually also full orientational information about the loops, right? And how they're evolving in time. And so there's a lot of rich information here, right? But since my background is in liquid crystals, we decided to focus on the dynamics of the structures, right? By the way, I should say, liquid crystals it's a one of the things that made soft matter, a commercially useful product. And this is a ubiquitous feature of liquid crystal. A liquid crystal nematic, actually the name nematic comes from thread-like, which means disclination, which the appearance is because of the disclination lines, right? And so this is kind of a defining feature of a nematic liquid crystal, but visualizing with such detail has not been done before, right? And so what's interesting is, again, so for example, you have this loop, and these loops can simply appear, one loop can appear out of uniformly aligned material. A loop by itself can also break up and contract and produce a uniformly aligned material. The other thing that you can do is you can have these extended lines, and they can self-intersect, and they can break off and produce a loop that can do all kinds of things. And the final kind of basic dynamical motif that we see is a loop that's growing in size and then eventually encounters this network and joins this three-dimensional system spanning network. Okay. And this is very different, right? Because from a two-dimensional sample, what happened in 2 dimensions is that the loop always appeared in pairs, right? You always had a pair of a +1/2 and a -1/2 loop appeared simultaneously. Here we have a single defect that appears simultaneously, and that would suggest that a topological charge, that this structure actually has no topological charge, right? Even though it locally suggests like there is a topology, right? So what I want to tell you in the last 10 minutes is, something that's somewhat specialized, but it talks about like how topology of defects in 3-dimensional fields is different from 2-dimensional fields, right? So this is the loop that, this is a +1/2 defect that I've shown you before, right? This is a core of a defect. If I go around this defect, I can take this rod, and what I can do is I can rotate a rod, and the rod rotates by 180 degrees, and it can rotate by 180 degrees because we have a nematic order parameter, so there's no arrows here. And this is your classical +1/2 defect, right? This is a +1/2 defect, and the way we mark it with this vector omega that's indicating the direction in which the rods are orienting, right? And then what you can do is you can simply say, right, so I go around this defect, around this core, around this vector omega, and I create this +1/2 defect. By convention, if the vector is pointing downwards, then you get a -1/2 defect. And again, in 2D, there is simply, without doing surgery, there's no way that I can take this object and transform it into this object, right? They're topologically distinct objects. Now in 3D, what happens? Again, this is just your -1/2 defect. And the reason for that is that the rods are forced to lie in the plane of the screen. If this is my 2-dimensional nematic, the rods are forced to lie in the plane of the screen, right? If I look at a 3-dimensional nematic, I could have a local, what appears like a disclination, right? That looks like this. The rod here lies in this direction, but then as you go around it, it escapes into the 3rd dimension, it comes back, right? And so again, what the crucial difference is that here, the rod is no longer confined to the plane of this interface. It escapes in this dimension and then simply comes back, okay? And the same way, and these are known as local wedge dislocation and twist dislocations, and these are known as local wedge winding character. And now an interesting thing happens, right? Is I can start with a wedge dislocation, and I can continuously rotate this vector omega. And as I'm rotating this vector omega, I go from something that looks locally like a wedge. You can see that these rods are twisting out of the plane, and they go back in this direction. I come back to the twist of the opposite character of a -1/2, and I can continuously simply go between a +1/2 twist and a -1/2 twist. Okay, that you cannot do in 2D. And that's also shown over here. Right, so you start with a 1/2 wedge that is familiar from 2D. You go into a twist, you go to -1/2 wedge, and back to the +1/2 wedge, right? And then if you close this thing, what you see is you create a disclination loop, right? Now, there's a lot of different disclination loops. Some of them can have a net topological charge, but some of disclination loops in 3D can have no net topological charge. And the two particular families that are important in our case are the mixed wedge twist loop and a pure twist loop. A pure twist loop is simple to explain, right? I have a local twist character, and you just close that into a loop, right? But you can see that this vector omega and this reference vector are uniform. So what I can do is I can simply contract this wedge twist loop into something that disappears. This is topologically neutral, right? The same thing happens over here, right? Here the omega vector is pointing along the loop, right? So in this case, I have a local wedge character, and here the vector is pointing against the loop, but along the tangent of the loop. And so I have a local wedge character again. But here I go through the twist character, right? So we've done an analysis, right, and I think about 300 loops. And again, there's many different loops that are possible in three-dimensional nematics, and you do see them in equilibrium nematics. But out of 300 loops, we've only found that all the loops that we see have no net topology, right? And they are a family that uniformly interpolates from mixed wedge twist loops to pure twist loops. And actually, the pure twist loops are somewhat more prevalent than the wedge twist loops. This is, liquid crystal. I mean, this was great for me because I always try to avoid learning about this. But once you do research on it, you can't not learn about it, right? So this is consistent. This structural analysis is actually consistent with the fact that these loops could just contract from zero and grow from zero uniformly. Okay. All right. So what I did is we assembled an imaging composite 3D nematic. We were able to image the structure of topological defects in 3D nematics and the nucleation mechanism of twist wedge and pure twist loops. Okay. And so again, that's very different from two-dimensional active nematics that I've talked about, right? In a certain way, what happens in a three-dimensional active nematics is you get a bend instability. The bend instability gives a local +1/2 and a -1/2 character, but these remain permanently bound by this twist character. And so they are growing apart, but as they're growing apart, they cannot break, right? They're topologically protected in a certain sense until there's a topological event. There's a twist loop that we also see nucleation of, and that's not in accordance to the normal scenario. It's actually unclear why the twist loops nucleate, but we believe that the nucleation of these twist loops, pure twist loops, is just driven by the background flow. The background flow is sufficiently strong to drive the nucleation of passive loop. Even if you take a passive liquid crystal, you put a shear flow, you can nucleate defects in them. All right, so the last thing that I want to say, and that's what we are working on right now, in principle, this is a material, and in this material, you have two fields. You have an orientational field that's described, that I've just talked to you about, and this length scale of this vector field is pretty confined, and it doesn't matter if I change the geometry size, right? So if I plot the orientation correlation function, it decays, and the decay length scale is of the order of tens of microns. Okay. On the other hand, what we've just started looking at is the interactions of this orientational field with the velocity field. We also have velocity fields, right? And so you can measure the velocity fields, and you can see the structure of these velocity fields. And you can see that the structure, at least the length scale over which this velocity field decays, is much, much larger than the structure of the nematic director field. And that's something that seems to be different than what's happening in two-dimensional active nematic. And I think it's interesting to think about how these defects give rise to the structure of the velocity field. What seems to happen is, and we have some preliminary evidence, is that actually the length scale of the velocity field of the velocity field, right, diverges with it, is determined by the system size. Again, that's a nice thing about our system, right, is you can plot the velocity correlation function. This is the most confined system. I think this is a 100 micron system, and you can see that the length scale is of the order of 100 microns, right? And so what we do here in the system is we start systematically increasing the confinement size, and we've gone to about 4 millimeters, right? And you can see that the length scale of the velocity field increases linearly. It's unclear if it's actually saturating here, because our box size is also of the order of 4 millimeters. And so what, so now we are doing, like centimeter size samples and see whether there is, I would love to have like, an active fluid in a smart active fluid, sell them in New York for a lot of money. Okay, so that almost brings me to the end, right? And so what I've talked to you about is, a new model system of a 3D active nematic liquid crystals. It's characterized by the velocity and nematic director fields that can be simultaneously measured on millimeter scale. The structure and dynamics of the orientational field is dominated by the dynamics of topologically neutral disclination loop. Again, one could imagine different scenarios in which you create pairs of disclination loops of opposite charge. We never see that. I think there's an interesting question whether there is an intrinsic length scale of the velocity field and how the velocity field interacts with the orientational field in 3D. And that's something that we are working on in the future, right? And so with that, I would like to thank you very much for your attention.
I've got a quick question about the loops.
Yes.
So if this was a system quenched from thermal
yes.
equilibrium,
yes.
They would try to minimize the surface, the
yes.
line length energy,
yes.
and so they would shrink like 1 over the square root of time.
Yes.
Motion by mean curvature.
Yes.
Does that still apply in this case or not?
No, actually,
right?
It doesn't, because you have active stresses, right?
Exactly,
and so then if you have this +1 distortion, that creates an active stress that wants to extend the loop.
Right.
But and so
So what is the prediction for how these things should?
Well, we don't have it yet, right? But and one of the problems with our system is that very quickly these loops don't live for very long, right? It's a very dense system. Their lifetime is very limited, right? So what we are trying to do right now is start with a point hedgehog defect, for example, in a droplet, and then nucleate a single loop and see what its dynamics would be, to really get one loop and see its dynamic and how it's correlated with its local structure. But in general, we'll see that if a loop is collapsing, the comet head is pointing inwards, right?
Yes.
If the loop is expanding, the comet head is pointing outwards. But that couples to the other advective fields, and it's not a general rule, but it's something we need to study in the future. I mean, right now we perhaps, the issue is that once the loops really start contracting, then that's when you have a lot of gradients in the director field, and that's when the dynamics is really fast, and our temporal resolution yet is not good enough. So we have some ideas of how to improve the temporal resolution to really study, when those singularities occur.
Thank you.
Yeah. Yes. No, it does not. It does not seem to. I mean, we haven't done an exhaust. I'm not going to publish this tomorrow, but it seems that the structure of the nematic field is well defined, and it doesn't depend on the confinement size. But the hydrodynamic fields, they increase with the system size. I don't know if there is, a certain length scale above which things get cut off, but we are in a good situation where we can make large samples.
So, I mean, this being absolutely not my field, I see actually a lot of interesting analogies with magnetic field behavior. Like, for example, what you're showing here looks a lot like flux freezing, whereas these like tubule structures, when they reconnect, and they seem to drive the fluid to more like chaotic topological features. This actually also has a really nice analogy with turbulent reconnection, where magnetic field lines, when they reconnect, they can drive additional instabilities and turbulence in the fluid. So
I mean, I'm actually trained as a physicist and don't know anything about turbulence. That's why I'm, you don't have to know anything about fluid dynamics at all. So I'm here to be educated, there, right? So. I would love to, if you can point me to literature to read. I mean, I would be very happy to.
There's a lot, yeah. So maybe we can
Yes,
discuss,
yes.
Yeah.
Well, I have one. I have one. Maybe I have one thing that I would like to raise about the discussion, or is that okay? Or so one thing that we are thinking about, and there's a group. This was not us, but there's a really talented grad student from Caltech who made these motors. And these motors, when you shine light, they dimerize and they generate an active stress. Okay. And then when you take the light off, these motors fall apart and the active stress disappears, right? And so now this gives you a unique opportunity to actively interfere in the developing dynamics of a chaotic system, right? So for example, here, we shine light and you generate locally an active nematic, and you generate locally, and so you have spatial and temporal control of the parameter that drives your dynamics, right? And so we are developing the system, because, the group from Caltech was great in sharing these materials. And the question I have that I think has a broader relevance is, I'm looking for control mechanisms, right? So for example, there is this dynamics over here, right? So in this case, the microtubules are now confined and they go in one direction, right? And every hour, these so what you'll get is this defect nucleation, and this is gonna continue, right? And every hour or so, it's gonna switch direction, right? And now my question is, I want to interfere in the dynamics of the system to make it actively switch direction, for example, right? And I would like a protocol that would tell me, okay, this is when I should shine light, and this is where I should shine light, and this is for how long I should shine light to make it go in the direction I wanted. And I'm just choosing this because this is like a bistable system, right? And so I don't know if other people in other fields think about this, but I think this is kind of a unique opportunity now where you can actually play with these fields locally, right? And that's my discussion question that I'm thinking about, right? So if somebody knows how to do this, we can test it in the lab. But I think that goes into control theory and engineering and all kinds of things I know nothing about.
on this, I mean, again, an ignorant question. I mean, how much of this depends on this species of virus or bacteria that you're studying? I mean, there's obviously what's driving these?
I mean, this is fairly universal, right? I mean, you have these equations with the orientational order. In this particular case, there's some tendency of the of the microtubules to accumulate on the side. But this is a fairly generic feature.
So it doesn't matter what kind of.
No, I mean, people see similar dynamics. You can confine cells in this geometry, and you'll see somewhat similar dynamics. You can shake granule rods. They'll do the same thing. I mean, that's kind of what's nice about, you take like something as dumb as a rod or as smart as a cell. I mean, and they do the same thing, right? I mean, so that I think somewhat justifies writing these equations of hydrodynamics that Sriram started a long time ago. And I mean,
but I mean, in some of the movies, the driver ends up being some sort of food, some sort like the biological driver of the motions of the swimmers.
No, but this
in some of the talks we've seen, right?
I mean, you could always have biological cues, but in this case, there is no biological cues. These things just land on the surface and move along the surface, and it's very. I mean, actually, biologists. There's a whole community of single molecule biophysics. For 20 years, they've studied how each individual motor moves, how many steps it takes, what is the force that So we know the microscopic ingredients really well, actually. And the challenge is to bridge the length scales. But I'll shut up now.
Something like this has been observed with bacteria and
yes.
So they do switch.
Yes. I mean, it's a very universal feature, right? But what I would like to do is be able to control it, right? So switch it on demand, or just to But I don't know how to do that.
Neither do the bacteria people.
Bacteria. Yeah, maybe bacteria.
So I had a question. Is there any hope of getting a system with polar order? I mean, there are other, non-liquid crystal systems, but even the bacterial systems, even though the bacteria are individually, they have a direction, the macroscopic state is always kind of nematic. Is there?
I mean, so what would be a quintessential polar order? I mean, instead of a flock, right? Or
that's all.
On a microscopic.
Yeah. Flock, but under confinement, it'll be a circulating thing.
I guess, I mean, the polar order is Andreas Bausch's thing on a 2D, the motility assay, right?
So that one is, that's right.
Right. Yeah. But, again, that's kind of an inherently 2D thing.
Right.
So, but yeah, if you had polar order, then you would have hedgehog defects and a different class of defects,
right? Okay, I think I'll just take over from here. Yeah. And I'm very happy how this discussion has already started, because I thought, and that's why I wanted to put up a couple of movies to get you even more riled up before dinner. There's, I think, two topics. One that we discussed a lot, which is that somehow in many biological systems there is this transition to a turbulent state, and now the idea is that there's energy and vorticity spectra which are consistent between different systems. And here I bring up on the far end over there, that is Zvonimir's system, which has this property. On the left-hand side, what I'm showing you here, this structure here, that's the mitotic spindle in a human cell, which pulls chromosomes apart. And what Christina Hüsken has done is she just kills off one of the motors of this thing. And what this thing transitions to is basically something like this, which I would call, or which they call turbulent spindles. So this is just in a living cell. We now just killed off one of the motors. We went from a perfectly controlled state to something that if we now intuit that what we have been saying throughout the day is correct, probably has a lot of the statistical properties of all of these active turbulent systems that we have. So there's, I think, two things which we want to discuss. One is how much does this fact that there seems to be some universality emerging help us? How much can we learn from the astro people? How much does it make a difference that in our case these are nematic field lines driven by some kind of biological fuels versus, let's say, magnetic field lines and some gravitational driving, or elastic stretch lines in some shared system, how much does this matter? How much does this put us in a different universality class? So is there a blessing to universality? Do we know much more about these systems than we realize by just stealing from other communities? On the other hand, what people like me want to understand is really this structure here, which is a very well controlled one. So now what you have been telling me all day is there is universality. So no matter what this thing is, if I just perturb it a little bit, I go to this state, which always does the same thing. So how do I can actually learn about what keeps this guy very controlled and able to do useful things for our survival versus going from here to there? So with this, I hope that some people here have ideas with this discussion. I think it's my privilege to just ask questions and hope that someone.
You didn't tell us about the one with the top.
Yeah, I forgot the one. So the point that I wanted to make here is, so this is also a movie from the Dojik lab. This is a different type of kinesin. The difference between this system here and this, it's a different type of kinesin, and there is no PEG, which is the polyethylene glycol, which puts microtubules together in this system here. So now one thing that we can sort of see in the in vitro systems too is that there are systems like this, which, so what the black stripes here are just bleach lines that you see moving very slowly, gently, as I would say, in a controlled, predictable way. There seems to be a way to go from a very controlled state, which I would say is more in the regime of real biological systems, to this turbulent state. But neither in the in vivo system nor in the in vitro system do we really understand this transition. So that's just another point that I want to make, that we have a question about how to control turbulence that we also probably as a community want to address.
I mean, I think what was interesting is Dan actually analyzed the length scale of a spindle, right?
Yeah.
And you can get the active length scale, and it just right above.
Can you use your seat mic, please?
Oh.
The other one.
So what Dan found is that, that length scale is just a bit larger than the spindle size, right?
Yeah.
So the biology must know when it's constructing this machinery, it must know what is the size of the spindle it can build and survive, right?
Yeah, somehow evolution seems to be able to avoid falling into the universal class that we're excited about as a physicist.
Yes.
And do something very specific. And maybe another system that I should bring up in this system, in the morning we heard from Jinghui Liu about row oscillations, which, as far as I understand, in the wild-type system also don't do the beautiful chaotic dynamics that she showed us, but do something much more controlled. And only once you overexpress activity via some, I don't remember what protein you played with, do you get into this generic class again. So,
Have you tried XTCK2 in the big system rather than KC1?
Yeah, I mean, it will under certain conditions go into the turbulent state.
Can you use your seat mic, please?
Okay.
Yes, it will. We tried it, and it does go into turbulent state under certain.
I just had an insider question. But what was the motor that was taken out here, and what does it do?
So in the in vivo system, there's two motors that are important. One that is kinesin, which is similar to the motors that are used in the in vitro system, which slides microtubules past each other. There's another one which is called dynein, which is in this system clusters at these pole structures here. And what they did, and that's the difference between the two movies, is that they either kicked out a factor that tells dynein how to find the minus ends of microtubules, or they just killed off the dynein. Then you go to the turbulent state. So in some sense, this is less active than this. So that's the... Yeah, the other interesting question, I think, beyond the control in biology, that maybe people have some ideas about is, if we study this state here, how similar do we now, after this discussion, actually think is this to the astrophysical and other examples that we have seen?
So just a thought on that is, I mean, obviously the statistical description that in the Monday and Tuesday talks were really pushed in terms of like the 2-point statistics and higher order statistics. And then today also seeing in some of the talks, like people were trying to look at scaling laws. I think that would be the avenue to really push further in terms of comparing statistics of turbulence in these different fields. And actually another thought that just came to my mind is that, from the machine learning side, I don't know, this may be a question for David. Has anyone done, just a simple classification model where you give it a whole bunch of simulations or whatever of turbulence? This is turbulence versus this is not turbulence. So like cat, dog, except turbulence, not turbulence. It'd be really interesting to just feed a neural network different images of this is what we call turbulence and then a bunch of things we don't think are turbulent. Well, let's see what the neural network says. I don't know. I think that would be kind of a cute thing. Well, that's what I want to get us all to do this week.
Yeah, maybe that's a question. Is this turbulence, in the opinion of the people that don't work on biological turbulence? We seem to have made up our mind that we want to call it that.
Could you use your seat mic, please?
So what does many mean in this context?
For us, we measure it as a Reynolds number. So it's usually when it's 1,000, it's not many. When it's a million, it's many. But it's empirical, right? And you can also, you need to compare it with something, right? So you can call turbulence something which has equilibrium, but your system has no equilibrium, right? Yeah. There's no thermal equilibrium. There's no Gibbs. There is no Gibbs state here. Yeah. So you use turbulence just because it's a nice word, or you want to say something about it which is different from what we are using, right? Yeah.
I was going to comment, maybe we shouldn't need to worry about terminology, rather worry about what's driving commonality, the mechanism, or the physical science, right? So one thing we can think of is sort of nonlinear dynamics, which have is well-defined subfield of physics, and it includes like fractal and chaos, probably turbulence too in general sense. However, they go beyond each system. They do the abstract way. I'm not really expert of that, but if you read the physical regulator, there's always a section about nonlinear dynamics. It's not the top one, maybe second and third one. You can look at that publications in general. They're trying to, self-organized criticalities beyond that particular field. And they go beyond the specific systems by trying to abstract the behavior of the so-called nonlinear system. And then we can quantify the so-called the operation or operator. And maybe we can come up to the high level kind of commonalities, not just the bio and the astral in general, and the complex system, how they behave. And I'm just trying to push out that one idea people can discuss about. It doesn't mean I know better than you, just observing it. Yeah.
Is there any other perspectives on this? Yes.
You several times said there is some universality. There is something which is similar between this. Can you elaborate what kind of universality in this active media system you see?
So one argument that we have heard, and I think it is not clear that this will experimentally bear through yet, although there are strong indications for it, is that if you measure cascades of vorticity and of kinetic energy in these types of systems, this one specifically, then we saw an example about cells moving in an arena. We saw an example about sperm swimmers going to a roiling state. There's examples about bacteria. All of these seem to fall on a common spectrum as far as we know yet. I don't think that it is completely
spectrum.
Velocity spectrum and vorticity spectrum. Maybe Luca wants to comment on that.
So they were like kinetic energy and entropy spectra, and there are 4 exponents which seems to be consistent across different physical systems. I'm here, I'm sorry, but I cannot look at you and speak at the microphone at the same time. And there are these 4 exponents that seems to be consistent and found across different physical systems. And interestingly enough, these exponents are actually kind of similar to the one that you observe in 2D turbulence. So this is why I think actually the name turbulence is not that bad for this phenomenon, because I mean, if I remove the labels from the plot that tells you the power law, I challenge everybody to distinguish it from 2D turbulence.
How long is your power law? Like 2 decades, half decade?
Well, the power law, I mean, it's actually, so there is a peculiar scale in the system that is equivalent to the injection scale into the turbulence, and other than that is only bounded by the system side. So it's as big as you want.
I mean, empirically, you see a power law, right? Not what you think about, you know about your system, but you see a straight line in a log-log plot. How long it is? How many decades?
Several.
In velocity?
5, something like that. I mean, so the most recent simulations about that have shown the power law convincingly for several decades. I mean, I can show you these.
Yeah, so there is strong indication that there is some kind of shared features between many microscopically very different systems beyond them just looking similar to the eye. So that's so really one of the questions to me is then, if we know that there is this, or if we can convince ourselves that there is shared properties between these systems, in what sense does the microscopics, can we still learn about the microscopics? Because in contrast to other fields, I think in biophysics, we are sort of in a scenario where we want to reverse engineer. We want to do these experiments to learn about the functioning of a cell, about the cytoskeleton, because in many ways, Zvonimir argued that we know about the single molecule properties of motors very well. What we do not know at all is what happens if we put 2 or 3 of them, because they interact in complex ways. And what they do in such a system and how this relates to what we measure in single molecules experiment, it's really unclear. So one of the questions for us, I think, is also how do we use these types of experiments to really learn about biology? What is washed out? What is truly common, and what do these teach us about how life actually works, how we do interesting things?
I have a comment with the previous question when we talked about spectrum and amplitude. Can you say that the amplitude of the spectrum was determined by the flux of the entropy or energy?
I have to give this question to Luca again, because I don't know. There was a question about the amplitude of the spectra and how they are determined.
Can you say that they're determined by the flux of energy or entropy?
Yeah, so this is the question that we were asking, also we were discussing at lunch. I mean, there are clearly spectra, so there are clearly multiple scales involved. It's unclear that all these scales are somehow activated at the same time, or there is indeed the flux of something. And we were discussing here, and I think Nigel wants to say something about it. It would be great to find a way to determine this.
Another question, was the spectrum isotropic or not?
So, so in turbulence, the way you can measure this is by looking at essentially third-order structure functions that tell you about, say, energy transfer or vorticity or whatever, entropy or whatever. So has anybody in these active matter experiments looked at third-order structure functions or third-order correlation functions?
Has anyone looked at third-order structure functions in these experiments?
Because not just you have a power law, it's just that you have a power law and something is going from one point in K space to another point in K space. That's what we think was happening in turbulence. Essentially, it's a state of PT symmetry because you have a flux.
Do you have a cascade or something like that?
Yeah, in a sense, we in turbulence never say cascade looking at spectra. Cascade for us is a third moment.
Exactly.
Spectra, just there are many systems for which spectra in equilibrium and in turbulence look exactly the same. Okay. Cascade is a third or fourth moment if you are,
So anybody who has data or big simulations, go and measure third order structure functions, and there's particular ones that you can construct that tell you flux, and then you should try it. There should also be some analog of the other question I have about while we're talking about this is there's a parameter in turbulence where you change the Reynolds number, you change the range of scaling, and I guess the answer in active matter is that you just change the size of the system. Is that right? Yeah. So then there should be some analog of 4/5 law. Have people looked at that?
So no, I was just going to say that I'm not sure all the analogies go through in exactly the same way, because the equation of motion that you're talking about, first of all, the object that's most like a velocity field with inertia is actually the polar order parameter. And the polar order parameter isn't a Galilean invariant variable. It's got a fixed scale. If a polar order parameter is a fixed length, there are many important differences between the hydrodynamic equation for the order parameter and the equation for the velocity field. And in the experimental regime where you measure the velocity field in these systems, the velocity field's inertia is totally unimportant. So, I'm just cautioning against uncritically saying that this is the way you want to do it. It'll be useful to look at flux vectors, definitely, both for polarization and for velocity field, contrast between the nematic and the polar case, things that I think these are good projects to start on. I thought you guys, Johan, you had done some things on this or not? Yeah, I thought the data range is very modest. Yeah. Right. Right.
I don't actually know why you want to take a problem where you're making a lot of progress on and mapping it onto a problem where no progress has been made.
That's a good remark. If inertia is irrelevant, the third moment is not a measure of energy flux, and you need something different. It's more something like elastic turbulence, namely something in which all the instabilities are not inertia driven. They are driven by memory which comes out of different parameters. And so if there is if you really want to ask what cascades were, you cannot say that you have an input on a scale of a single, bacteria or in a single rod like you. Of course, you have an input of energy on the scale where they start to be correlated and making correlated motions. That's where you actually you input energy in the macroscopic mode, and it's totally different, of course, from. So you need to kind of just write an energy equation, take 2-point density, and see what gives you flux.
Is there any further comments on this? Otherwise, I think we are out of time. And